students. In this lecture, let us learn about membrane transport. Let us start with the introduction. The phospholipid bilayer of the plasma membrane is impermeable to ions and water-soluble molecules such as glucose and amino acids. However, the property of membrane is modulated by the transport proteins associated with the bilayer, facilitating the transport of the ions and water-soluble molecules and making the membrane selectively permeable. The plasma membrane of different cell types contains different sets of specific transport proteins for the transport of specific ions or molecules across the membrane, since different cell types require different composition of these low molecular weight compounds. For example, epithelial cells lining the small intestine transport products of digestion such as glucose and amino acids into the blood, while those lining the stomach secrete hydrochloric acid into the stomach lumen, and therefore these epithelial cells have different sets of transport proteins. Let us now see the physical and biological transport processes across the membranes. The main transport processes that move molecules across membranes are diffusion, pinocytosis, transcellular transport, and carrier-mediated transport. Let us take up the first process, that is diffusion. Diffusion is an important mechanism for the movement of molecules across the lipid membrane from an area of higher concentration to an area of lower concentration, and the rate of diffusion of solute is directly proportional to the difference in concentration between the two areas. The diffusion of molecules across the cell membranes may occur directly through the lipid bilayer for lipid-soluble solutes or may occur through specialized channels in the membranes. The next one is pinocytosis. This is another process of membrane transport which involves engulfing of extracellular fluid by invaginating cell membranes. The invaginating cell membranes form a vesicle which later separates from the membrane and may move through the cytoplasm to release its contents on the other side of the cell by means of an evaginating process called exocytosis. The third one is the transcellular transport. Such type of transport usually takes place across a layer of cells and occurs through carrier or channel molecules present on the luminal and antiluminal sides of the cells. Transcellular transport may be facilitated diffusion or active transport. In transcellular facilitated diffusion, the membranes on opposite sides of the cell usually have similar carriers and solutes are transported along their concentration gradients. While in transcellular active transport proteins are present only on one side of the cell and the other side of the cell usually lacks active transport system, giving different properties to the plasma membrane of the two surfaces of the cell called cellular polarity. Such type of arrangement leads to the accumulation of solutes within the cell by active transport through the transport proteins on one side, and the solutes leaves the cell from the opposite side of the cell through a channel or facilitated transport. The next is the carrier-mediated transport. As the name suggests, carrier-mediated transport involves the transport of molecules across the phospholipid bilayers with the help of transport proteins. The membrane transport proteins can be grouped into three major classes. The first one are the ATP powered pumps. These pumps are ATPases or protein pumps that utilize the energy generated from ATP hydrolysis to transport small molecules or ions across the membrane against a chemical concentration gradient or electric potential in an energetically favorable manner. The process is referred to as active transport. ATP powered pumps are responsible for maintaining low calcium ion and sodium ion concentrations inside the cell compared to outer medium in most animal cells. They also maintain a low pH inside lysosomes in animal cells, in plant cell vacuoles, and in the lumen of the stomach. There are four classes of ATP powered pumps: P, F, V and ABC or ATP binding cassette classes. Among these, the P, F and V classes transport ions only, while the ABC class transports small molecules. Let us see the P class pumps. Structurally, the P-class ion pumps consist of two identical catalytic alpha subunits that contain an ATP binding site. Most of them also have two smaller beta subunits with regulatory functions. These pumps are called P-class since the transport by these pumps is associated with the phosphorylation of at least one alpha subunit through which the ions are believed to move through. Examples of this class includes the sodium-potassium ion ATPases in animal cell plasma membrane, which maintain low cytosolic sodium and high cytosolic potassium ion concentrations by exporting 3 sodium ions for every 2 potassium ions imported. Calcium ion ATPases pumps of plasma membrane and specialized endoplasmic reticulum of muscle cells called the sarcoplasmic reticulum pumps calcium ions out of the cytosol across the plasma membrane or into the endoplasmic reticulum from the cytosol across the sarcoplasmic reticulum. Proton transports found in acid-secreting cells of the mammalian stomach that pumps out protons and pumps in potassium ions, as well as the proton pumps in membranes of plant, fungal and bacterial cells that generates and maintains the membrane electric potential. Next are the F-class and V-class ion pumps. The proteins of these two classes are structurally similar but are unrelated. Both F and V class pumps have many different transmembrane and cytosolic subunits and they transport only protons without involving a phosphoprotein intermediate. Examples of V-class pumps include the proton pumps of plant vacuolar membranes, lysosomes and other acidic vesicles that maintain low pH inside the vacuole or vesicles. On the other hand, the F-class pumps, also commonly called ATP synthesis, are found in the membranes of mitochondria, chloroplasts and bacterial plasma membranes, where they power the synthesis of ATP from ADP and inorganic phosphate by pumping protons from the exoplasmic to the cytosolic face of the membrane down its electrochemical gradient. Next are the ABC or ATP binding cassette superfamily. This class of pumps have a general structure consisting of 4 core domains, 2 transmembrane domains that form a passageway for transporting molecules across the membrane and two cytosolic ATP binding domains. Each ABC transport protein transports a specific single substrate or a group of substrates such as ions, glucose, amino acids, phospholipids, peptides, polysaccharides, or even proteins. Next, let us take up the channel proteins. These are protein-lined passageways in membranes that transport water or specific ions down their concentration or electric potential gradients in an energetically favorable way. Such type of protein-assisted transport is known as facilitated diffusion. The channel proteins transport many water molecules or ions at a time rapidly with a rate of about 10 to the power 8 per second. For example, potassium-specific channel proteins of animal cell plasma membrane that generates the resting potential across the membrane. Some type of channel proteins transporting ion remain open most of the time and they are called non-gated channels. In contrast, most ion channels remain closed most of the time and open only in response to specific electrical or chemical signals, and they are called gated channels. The next group are the transporters. These are membrane transport proteins that transport ions and molecules across cell membranes, usually one or a few at a time with an average rate of about 10 to the power 2 to 10 to the power 4 molecules. The binding of substrate molecules to the transporters is specific and it results in conformational changes in the proteins that allows only the bound substrate molecules to be transported across the membrane. Transporter proteins can further be grouped into three types: uniporters, antiporters, and symporters. The uniporters transport only one molecule at a time down a concentration gradient across the membrane. For example, uniporters that transport glucose, which is the GLUT1 transporter, or amino acids into mammalian cells. On the other hand, the antiporters and symporters carry out the transport of a type of ion or molecule uphill against its concentration gradient, which is usually energetically unfavorable, coupled with the transport of another ion or molecule downhill its concentration gradient, which is an energetically favorable reaction. Although these transporter proteins are often referred to as active transporters, they do not involve hydrolysis of ATP during the transport of molecules. Therefore, these transporters are also commonly called co-transporters since they are capable of transporting two different solutes simultaneously. Such type of transport is also called secondary active transport since the co-transporters use the energy stored in an electrochemical gradient, unlike the ATP pumps that use energy from hydrolysis of ATP. Let us now see the conclusion. Although the lipid bilayers of membranes are highly impermeable to most polar molecules, many small water-soluble molecules are transported across the membranes with the help of various membrane transport proteins. These membrane proteins help in carrying out both passive and active transports across the membrane, and they also play a major role in maintaining pH inside cell or compartment or generating membrane potential. Thank you.